Hi guys.
Hi.
Hello, Miss.
Hi. So everybody's here.
Yes.
So we are missing Lou, is it?
I'm here.
Hey, which one are you? I see two host name.
I log in using Cole's link because I did not find the email with the Zoom link.
Oh.
Is
Okay.
Cole able to log in or is he in already?
I'm not sure. Hi Ko, are you here? Lou is from Pamer, Indo. In case you want to check again.
Mary Indo, okay.
Yes.
Yes. Maybe I should log out and use back my own link, yeah, in case
Yeah, that will be
lucky
better.
person. Yep.
Yeah.
All right. See you then.
Yeah. Hi Lou.
Okay. I'm in again.
Yes. Now I see you.
Yeah.
So, okay, let's see. So we have a Q&A box.
Yep.
And then we also have a chat box.
Right.
So, okay, maybe one of us should monitor
One of them.
Yeah.
Yep.
So would you like to monitor the Q&A box and I monitor the chat box?
Okay.
Mutia,
Yes, Miss.
how about you? Because, customer might ask questions for sales related from chat box and from chat and Q&A box. Can you look at both?
Yes, Miss.
All right. Okay, cool. So we start sharp at 1:55.
Yes.
All right, cool.
And also, Miss Aswa, I will share a form in the chat box.
Okay.
For registration of the attendees.
Okay, all right. Actually, in Co's presentation, there's a QR code as well. So in case if the customer has further inquiry, they want to know more about the products or they want to be contacted by you later, they can scan the QR
Okay.
code. Yeah, and we will send over everything that we have to you after the discussion, after the presentation.
Oh, okay. All right. So the Q&A is after the presentation, right?
I think so. Yeah.
If there is any question about the seminar product or whatever in the chat box or Q&A, I will try to answer that. If not, those unanswered questions will be dealt with at the end.
Okay.
By Co, yes. Are we hearing anything from Co?
Let me check with him. But he's here anyway.
Yeah. The recording has been started, is it?
Not sure. Need to check with the host.
Because I saw pause or stop button at the bottom, but there is no start button.
Yeah, we already start recording.
Okay. Thank you.
All right, cool.
Okay.
Thank you. So when can we share our slide? A question to the host. No?
No response.
Hello, sorry, hello.
Yeah. When can we share our slide?
Yes, please.
We can share it now?
Yes, please.
Okay, thank you.
Dan wait baru masuk wait. Oh siap udah masuk ya. Dan baru masuk. Coba Pak Budiman ya. Iya ada itu ada kamu ada. Ada ya.
Oke, baik. Selamat siang semuanya. Selamat bergabung di webinar PT Infinity Bioanalitika Solusindo. Oke, mungkin sebelum kita masuk ke topik utamanya, sedikit saya ingin memperkenalkan mengenai perusahaan kami. Jadi perusahaan kami PT Infinity Bioanalitika Solusindo ini merupakan distributor alat-alat laboratorium dan kita juga merupakan sole agent dari Eppendorf. Oke, pada kesempatan kali ini kita akan melakukan webinar dengan topik cell micromanipulation in agriculture, animal breeding, and biomedicine yang akan disampaikan oleh Mr. Kwan yang merupakan Eppendorf Regional Marketing Manager. Oke, dan untuk nanti kalau misalnya peserta ada yang ingin bertanya, bisa langsung bertanya di kolom Q&A. Oke, mungkin untuk mempersingkat waktu, saya persilahkan Mr. Kwan untuk memulai webinar kali ini. Terima kasih.
Thank you, IBS, for the very nice introduction. I roughly understand what you're talking about. Let me just share my screen. Can you all see my my screen?
Yes.
Yeah. I'm going to choose the virtual background. Do you see the virtual background?
Not yet. It's your green background.
Okay. I think it might not work with my virtual background here.
Oh, okay.
Okay.
Yeah.
All right. Without further ado, thank you very much for joining this webinar. So as introduced earlier, the title of this webinar is Cell Micromanipulation in Agriculture, Animal Breeding, and Biomedicine. So my name is Kwan. I'm from Eppendorf, Regional Marketing Manager for East Asia Pacific and Japan. Okay, just some housekeeping rules about this presentation. Please kindly mute your mic during the presentation to reduce background noise. And if you have any questions during the presentation, please type in the Q&A chat box. My colleague, Ms. Lu and Ms. Asfa will help to answer the questions due to the interest of time, so that it can save us more time. And anyway, at the end of the presentation, we are going through the questions that you might have in the chat box. And during the presentation, if you see at the right corner of the slide, each slide, there's a QR code, and also there's a URL address. So if you have any feedback or any questions you would like to ask, or maybe you would like to meet with our representative, arrange for a demonstration, anything, you can scan this QR code, it will link you to a Google form, and for you to fill up the particular and also the question that you might have. And I'm very pleased to say that we are giving up 4 mystery gifts for lucky draw winners who fill up this feedback form for us. So without further ado, let's begin with today's topic. I'm going to start with the introduction of the technique. I would like to introduce what is a micromanipulation or microinjection is all about. And then we are going into the application of microinjection technique. And then we'll introduce Eppendorf microinjection system, okay? Micro cell manipulation basically is a technique that you perform under the microscope. So because you're manipulating the cells, you need to use a microscope to see the cell. So we can't use a hand to manipulate the cell because we are moving from one cell to another cell. So we have to rely on an instrument. So, and the instrument is called a micromanipulator. So what is micromanipulation? Cell manipulation could be, for example, picking of the cell of interest that you want to inject. It could be stem cell or cancer cell or whatever cell that you want to manipulate. And then you perform the cell transfer. So you inject a particular cell into the oocyte or egg or embryo or blastocyst, depend on what experiment you want to perform. Or most of our cell lines, they are in adherent form. So it means that they stick on the dish, so that you can also, with the DNA or sample that you want to inject, you load into the very fine capillary, and you inject into the cell directly. Depends on what kind of experiment you want to perform. All right. And how it is performed, basically, of course, you need to prepare the cell under in a dish, a petri dish, and then you place under the microscope. And then enlarge the image under the microscope. Then this is where the micromanipulator comes about. So you have a microcapillary. It's just like the injection. You need to use a needle. So the microcapillary is a glass needle, very, very fine, where instead of inject into our arms, our body, but inject into the cell. And in order to move the microcapillary, then we need to rely on a machine called micromanipulator. So the micromanipulator can move in a very fine and slow and defined movement, so that you can move from one cell to another cell to perform the work. All right, and the micro injector to give the pressure. It's just like the syringe to, with a plunger to give the pressure. So basically, this is the concept of micro manipulation. It's all about, or we call it a micro injection. So they're actually basically the same. And some advantages why people perform micro injection technique. First and foremost, with this technique, there's no limitation of what you can inject into the targeted cell. All right. So this technique is also very efficient because you inject directly through the very fine needle to deliver the sample of interest. And if you tag together with a fluorescent dye, immediately after the injection with the fluorescent microscope, you can see whether your material is inside the cell or not. For example, as you can see from the picture here, here there's a fluorescent of the sample. So when you inject together with the dye, then immediately you can check whether you have successfully injected the sample into the cell. And the other advantage of the technique is you can decide what area you want to inject. You can inject either into the nucleus of the sample, or you can inject into the cytoplasm. So it depends on your goal, the goal of your experiment or research. So in terms of what can be injected into the cell with this technique, and this is one of the biggest advantages of micro-injection technique, there's no limitation what you can inject. You can inject DNA, RNA, antibody, oligonucleotides, or even dye, or even cells like sperm. And this is very popular for IVF center, where they inject the single sperm into the egg to fertilize the egg. Or cells, depends on your sample. Even the bacteria, microphage, and also bacterial cell that you want to inject, and stem cell and cancerous cells. And what some main applications for micro-injection techniques are to generate transgenic animals or transgenic organism. It can be plants or animal, or IVF, or for animal, we call it animal breeding, or for biomedicine production, to clone the animal. As you know, that very popular is the dolly sheep, is cloned using a micro-injection technique as well. And genetic disorder study. So transgenic animals can be used as a model to study inherited disease or genetic disorder. And for example, very popular now is they're using zebrafish to study the genetic disorder. So today we are just only going to focus on 3 applications. They are the transgenic animal production, animal breeding, and biomedicine. Now let's go into the application. What is transgenic organism? Transgenic organism is an organism whose the whole genome has been changed. So you can insert a gene from a different species to another species. So it has a mixture of a different gene. And this, why people produce a transgenic animal is basically mainly for animal, for human welfare. For example, to produce substances like proteins, they can treat certain diseases for therapeutic purposes, or as a disease model to study genetic diseases. And up to date, a lot of organisms has been produced as a transgenic animals. They are goat, mice, rats, pigs, cows, even like plants like oil palm, rice, soybean have been created. Okay, here I'm just going to give you one overview picture of how this the relationship between transgenic animal production, microinjection technique, as well as animal cloning. So if we start from the top here, you have a gene of interest you want to produce, maybe a certain gene encoded with a certain protein from human, and then you inject into the animal oocyte or the fertilized oocyte here via nuclear injection. So you can inject the gene of interest directly into the pronuclear to produce a transgenic animal. With the recent advancement of the technique like CRISPR-Cas9, this is a popular method. Of course, there are other methods to produce a transgenic animal, but this is a fast and a very popular method now. And here, that's how you perform the injection. You transfer the gene of interest into the pronuclear. So with the micromanipulator, you inject directly through a capillary with the gene of interest, inject directly into the pronuclear. So when the embryo developed, and then you transfer into the foster mother to carry and produce the fetus and the babies. And once you create it, because transgenic animal production takes a long time, it's not easy to produce a transgene that you want. So when you want to maintain this animal with the gene of interest that you want with the transgene, you can perform animal cloning. So animal cloning is to clone an identical animal. So first, and you get the egg from the animal, and you remove the nucleus from the egg using microinjection technique. So this is a video to show how the nucleus is removed from this mouse egg. And this process we call enucleation. And the product of this technique is called enucleated egg. So once this egg is enucleated, it doesn't have the genetic information inside, then you can insert the nucleus from a different animal or with the transgenic animal that you want to clone into it. And then you use a microinjection technique. You transfer the nucleus that you want into this enucleated egg in order to clone an identical animal. And this is how animal is cloned, and also the advantage of a microinjection technique. I'm giving you some very commercial and province examples that people use a transgenic animal to produce the product. One example is in agriculture, and this is a publication from this group in Malaysia, Malaysian Palm Oil Board with association with University in Münster in Germany. So they use a micro-injection technique to produce a transgenic oil palm protoplast. So the objective of this study is to produce transgenic oil palm with higher content of oleic acid and with a modified oil quality, like have a higher content of stearic acid, palmitoleic, and also ricinoleic acid. And at the same time, it is also used to produce a novel product like a biodegradable plastic. Okay, now let's look into what they do with this experiment. So they inject the DNA into the protoplast of oil palm, and at first, because they want to check whether the gene of interest is inside the protoplast or not, they inject together with the luciferase fluorescent. So you can see there's a fluorescent. And then here you isolate the protoplast with the transgene and you culture it. After 3 days, you can see there's a green fluorescent being expressed in this protoplast to ensure that this transgene is being integrated into the oil palm protoplast. And some advantages are outlined in this publication. They find that compared to the conventional method they used for transgenic plant production, like bombardment or the gene gun and acrobacterium mediated method, with the microinjection technique is faster and easier to perform. And because they do not need to produce a lot of tissue compared to conventional method, with less than 0.5 gram is good enough already. And the other advantage is they can create a cleaner gene fragment, which consists only the promoter gene terminator sequence, which can be introduced to avoid transgene interference and produce a chimeric single cell eventually, which they want to avoid. They want to create a transgenic animal with a transgene and not the a chimeric plant cell. And when we go from plant to animal, there are a lot of examples that we can find that the people use transgenic animals. One example is AG Research. In 2017, they successfully produced their first transgenic cows. And this transgenic cows is able to produce more casein. Casein is a kind of a protein that is used to create, for example, cheese making. So they have a double level of kappa casein for cheese making and increase the calcium level. And this is a picture of how the transgenic goat looks like. And a group of scientists in China also have successfully produced a transgenic cow that can produce a humanized milk. That means that this humanized milk contains more lysosome, lactoferrin, and alpha lactoalbumin. And this is all these proteins contained inside the human milk, where it's very important to boost immunity and also prevent infection for infants or babies. In biomedicine, so the first FDA approved medicine that is produced by a transgenic animal is called Atrin. So this was produced in 2009 by a company, and this end product is called Atrin. Is used to treat anti-thrombin deficiency. It's a kind of a blood clot disease. Is a genetic disorder for the patients. So how this is created? Basically, they create a transgenic goat, and then from the milk of the goat, it has this human protein. They extract this human protein as a medicine. And the other example that is also FDA approved is called Kanuma. Kanuma is a drug from a transgenic chicken. So it's used to treat a disease with lysosomal acid lipase deficiency disorder. So this is another example of the end product. And one very interesting example that I'm going to share next is BioSteel. So it's produced by Nexium Biotech in combination with joint venture of US Army in 2000. So they produce an animal called spider goat. It's a transgenic goat that can produce spider silk in their milk. So of course, the goat doesn't look like like a monster here. They actually look like a normal goat, like a picture here. So with this goat, why they produce spider silk? Spider silk is first and foremost is human compatibility. Is compatible with a human and is 6 times stronger than steel. And they use the spider silk to create bulletproof clothing, aircraft and racing vehicles. And for medical purposes, they use the spider silk to produce medical sutures, meshes, and even artificial ligaments and tendons to treat a patient with ligament and tendon issue. And this up to date, they are still producing this product. For animal breeding, now we're going into the animal breeding with the micro-injection technique, and this salmon called Aqua Advantage salmon is a transgenic salmon that is produced using micro-injection technique. So with this salmon, why they produce this is compared to the Pacific salmon, they can only grow during spring and summer, and the size to reach an adult size, it takes a very long time, up to 3 years. But with this genome modified salmon with a transgene that can that have this growth hormone transgene inside the salmon, it can grow faster throughout the year. So it depends. Regardless of what season, they can produce anytime, and also it takes only 3 months to grow to an adult size compared to 1 or 3 years. The next example that I would like to share, and it's also very interesting, is a transgenic sheep. So they create a transgenic sheep to have a more wool production. So they overexpress the insulin-like growth factor, so that the sheep will produce more wool than normal. Okay, next I would like to share with you how can all this be done using micro-injection technique. I'm going to share with you the Eppendorf system for to support all this micro-injection technique. This is how the system looks like. So you need to have an inverted microscope and attached with the micromanipulator. So with this microscope, you have the micromanipulator. As you can see from the side, this is with a joystick to control the movement here. So it's just like controlling the machine or playing a video game. So you move the joystick, and then at the same time, this needle will move under the microscope. And our system, the Eppendorf system, is electronic system. Unlike the conventional system, they are using hydraulic system or mechanical system. Is electronic system, so that you can perform even a finer movement, and also faster control, and maintenance free. So unlike the hydraulic system where you have to fill up the oil to use the hydraulic pressure to move the needle, this one you do not need to fill the oil. You just plug in, plug and play. And it's very easy to operate with just a single joystick to control all the movement, the left and right, front and back, and up and down of the needle with just a single joystick. And we have, as you can see, there's a very minimal component that you need to control compared to the conventional system where there are 10 to 14 components to move different direction and different fast speed, low speed. They have different components. This one is all in a single joystick. And there's a control panel here where you can switch the speed here. And it's very ergonomic design, because for micro-injection, especially when you do not have experience, in order to acquire the skill and the technique, it takes a long time under the microscope to perform the work. And it can be very tiring if the design is not ergonomic. And just like nowadays, our our smartphone here. So electronic system with the smartphone, advantage is the software is upgradable. So whenever there's a new function or there's a new software, you can upgrade the system without having to purchase an additional device or the machine for a long run. So it's a better investment for a long run. And here I have a short video here to show what it means, how to control the system in order to perform the micro injection. So with a single joystick here, so you just move the joystick to the area you want to perform the work. And this is a microscope view with a small needle. So you rotate up and down to move the needle, and left and right to go to move the needle. And then there's a function where you can remove the needle away from the dish when you finish the work. And there's also additional, a lot of useful function. For example, this home function. So when you press this home function, when you want to change the capillary of fine needle, you just press this, the system will automatically move away from the dish, and you just change a new and clean capillary with a new gene or a sample you want to inject. You replace it, you press the same button here. There's a home button here. You press the same button. It has a memory function. You can go back to the previous position where you stop the work. So you do not need to find the needle from scratch from A again to make the work faster. And I talk about the ergonomic, it's very important. So here is very ergonomic design where you can place the arms. You can see the user here, they can put their hands comfortably on the control panel during the work. So unlike certain systems, they are having a hanging joystick where you have to hold like this for a long time. So it can be very tiring for a long working hour. And also this is portable. You can move and place the control panel according to your arm's length. So some people might have a shorter arm, some have a longer arm. So you can put it comfortably and very ergonomic for your entire work process. So eventually it can reduce the stress, not just the mental stress you have to perform the work and produce result, but also the physical stress. And there's another very useful, very interesting feature of this system is it has a memory function. So it can memorize the position where you perform the work, up to 3 different positions. For example, in the microscope here, there are 3 positions where you want to see the needle in the microscope and also the cell, and also do the injection work, and also where you separate the injected and non-injected sperm, the cell. So you can, with this system, you can memorize the position. At any time, you just press the button here to go back to the position that you want to continue the work. And also, it has the widest working range available in the market. That means that this is a microscope view. So you can move around this area. Let's say you have a lot of cells here. You want to go to this area to catch this cell. It looks better. This is the cell that I want to isolate. I want to inject. I can go here without having to move the dish. So the area that you can go is wider and larger compared to other systems available in the market. And as I said in the beginning of the presentation, is electronic system. There's no need to fill the oil. It's maintenance free. Just make sure that you pay the electricity bill and you just plug and play the user system. And it's upgradable with the software anytime. When there's a new function, there's a new software, the engineer can go to your place and just plug with the USB drive and upgrade the software for you. So basically, there's the end of my presentation about the micro injection. And if any questions, please go to the feedback form here, and you may scan this QR code here, or go to the URL here to ask question, or whether you want to inquire a product demonstration brochure, you want to have a discussion with our application specialist about the technique, how you can use this technique, apply onto your research and also your experiment, please go to this the feedback form, fill up the feedback, and then we will have someone to contact you and address your inquiry. And also, I would like to again mention that we are giving out 4 mystery gifts for those who are logging into this feedback form and provide the feedback. And we are going to have a lucky draw for 4 lucky winners. And we're going to announce and contact you if you are one of the lucky winners. Thank you. That's all for my presentation. Let me see if there's any questions from the floor. So we have a question from Shanti Putra. I see this system use a microscope. Is any microscope can use? To answer your question, yes. You can use any brand of microscope, and we just need to, we have a adapter for different brand of microscope, and also a universal adapter to adapt the microscope you're having. And also what microscope to use, it depends on your experiment. And there are different types like inverted microscope and also upright microscope. So if, for example, you are doing injection, like you want to inject prawn nucleus for the animal nucleus, then it's more suitable to use an inverted microscope. We do have the adapter for all kind of brand and models of inverted microscope. If you want to perform work like inject into a bigger organism, like plant cells or insect cells or worms, and then you need to use a upright microscope, or we call it dissecting microscope as well. Then you need to use a different kind of adapter. So again, also depends on what kind of experiment that or research that you want to perform. Then you choose the right microscope. We have adapter for all types of microscope. Do we have any more any questions from the floor? Now we are open for the floor to ask the question. You may unmute your mic if you have any questions to ask regarding the technique or the application. Okay, maybe you do not have any questions or at the moment, or you have some other question you want to ask. You can, you may fill in the feedback form and send us an inquiry. You can fill up your comment there, and then we have someone to approach you and contact you. The floor is open now for Q&A session, so you can either key in the Q&A box, or you can unmute your mic and ask the question verbally. Okay, we have another question from Muri Arisanti. How long it would take to build an animal model for cancer? Do you mean to build a transgenic animal for cancer research? Oh yeah, so that that is a very subjective question. It depends on lab to lab and also your experience to produce. So it could take 1 up to 5 years, and it depends on the lab setup also. So if you have experience doing this, create a transgenic animal, and also already have stabilized with the technique, then it could be very fast. So It's a very subjective question. Again, I can't really give a definite answer to that. However, that one of the key component to decide how fast it can produce that is depends on the how fast you can optimize the technique. One of the techniques is the how fast you acquire the micro injection technique to deliver the gene, and also how fast you can produce the DNA construct or the gene that you want to perform the transgene. So that actually depends on from one lab to another lab. But with the system is very easy to use. So that means that your personnel can upskill and also can master the injection technique faster. Then the faster you can produce the transgenic animal models. How long from another user, thank you for interesting session today. Is it any different between plant and animal treatment to do microinjection? Yes, definitely there's a different treatment. For plant cell, usually because it has the cell wall. So because the cell wall is very hard to penetrate, so with the transgenic method, this is the biggest challenge for plant cell injection. So you need to have a special treatment to remove the cell wall and inject into the protoplast. With the animal cell, it is easier. As long as you produce the cell with the right confluency under the dish, you can start the injection immediately. And also it's faster compared to a plant cell. Plant cell that you need to have a certain method to prepare your protoplast in the agarose, and the agarose level cannot be too thick. And because it's too thick, then you will block the capillary. So there are some optimization protocol for plant cell compared to animal cell injection. Does this microinjection need a special room like a biosafety cabinet? Yes, you might need, but not necessary. We have a lot of researchers, they perform without the clean room or not inside the biosafety cabinet. If you do, if the contamination is not that your concern, but some with the concern of the contamination, you can have a box that can cover the microscope and also the micromanipulator system. So that they do have this commercialized box that can cover this and to reduce and also to prevent the contamination. Is there any specific strategy or technique to get best result of transgenic animal in terms of controlling the capillary needle and control joint, every kind? Definitely there's the technique involved to give a best result. For example, whether you want to inject your gene into the pronuclear or into the cytoplasm, it depends on your experiment design. So some may be expressed better in the cytoplasm, some is better you inject into the nucleus. So then again, the question, the answer goes back to the kind of experiment and also the research and also the cell that you want to inject. And of course, injecting the technique plays a very important role as well, because with the longer exposure the cell outside the incubator, the lower the survival, and also the needle that you use has to be fine enough so it reduces the damage or the injury to the cell, and also the speed that you inject and also the pressure. So all this has to take into consideration and optimize and in the beginning of the experiment until you get a very good and optimum protocol and give a good result. If using another microscope such as bacteria or another micro such as bacteria or yeast, the method for injection is same like in the cell. So what I show you in the video is just some examples. So there are some other models like you want to inject into the yeast. If you inject into the yeast, it could be quite similar to like what I show you in the video. You need to have you prepare under the dish, and then you have a microcapillary and inject directly. And I have done the micro injection into the yeast, and it works well. And we actually have one customer in Indonesia actually doing that micro injection into the yeast, but I'm not allowed to say here the name here. And other microorganism, it depends, like as I said, if you're injecting into the insect cell or mosquito, then it will be a different kind of method. But if you would like to know more, we are happy to have a discussion, a more detailed discussion with you about your experiment and what kind of method, whether this method is suitable for you. Micro-injection technique is good, but may not be applicable for all kind of experiments. So I encourage to have a more customized discussion or meeting with you, so that we understand your research, and we'll be able to give a more customized answer and also the solution for you. All right, thank you so much so far for your questions. I hope I have answered most of the question, and you'll find this session interesting, and it can be helpful for your planning on your experiments or your work. So if due to the time constraint, we only have 3 more minutes left for this session. If you have a further question, do remember to scan the QR code and then address your question in the feedback form, and then we'll be in touch. Okay. Okay, I see there's no more question from the floor. So to end the session, IBS, would you like to say some final words before we end this session? IBS?
Okay. Yes, thank you, Mr. Ko, for your presentation. Oke, kepada semua peserta, terima kasih sudah menghadiri webinar kami. Dan untuk tadi beberapa pertanyaan yang sudah dijawab, semoga bisa menjawab pertanyaan Bapak Ibu sekalian. Kepada Ibu Muri, silakan menghubungi admin kami untuk redeem souvenir, dan juga Ibu Emilia untuk menghubungi admin kami. Dan juga kepada Bapak Abbas bisa menghubungi admin kami untuk redeem souvenir. Baik, mungkin cukup sekian dari kami. Terima kasih banyak atas partisipasinya. Selamat siang.
Terima kasih. Bye-bye.
Oke, bye-bye. Thank you, Mr. Ko.
Bye-bye. Terima kasih.